Good afternoon, students. Good afternoon, everyone. Yes, good afternoon. Good afternoon. Yes, we are discussing previous year question paper for 2023 Shift 2. Okay. We have already discussed first 60 questions. Remaining questions we will cover in this session. Okay. Is there any doubt in the last session? I hope we have given our 100% to explain each and every question. Okay. Be attentive in class and kindly interact. Okay. First questions of today's session. Yes, first question. What is not the query sequence in BLAST N? This we have already seen. What is not the query sequence in BLAST N? What will be not the query sequence? Whether nucleic acid or proteins? Yes, excellent. Ram Virat. Listen the question properly. What is not the query sequence in BLAST N? They are not asking about BLAST P. BLAST P is for protein. BLAST N for nucleotides, right? So BLAST N protein will not come, correct? Next, what is the most relevant use of BLAST? What is the most relevant use of BLAST? For BLAST, why? For what purpose we will use? This we will use for sequence alignment. Yes, primarily we will use for sequence alignment. We will use sequence alignment. Yes, very good. Sequence alignment. Universally required vitamin in tissue culture medium. What is the universally required vitamin in tissue culture medium? As we know, vitamin B1 is primarily required in plant tissue culture medium. Okay, plant tissue culture medium. Okay. The other name of vitamin B1 is what? Thiamine. Okay, thiamine is the other name. Yes, excellent. So thiamine hydrochloride is other form. This remaining things is what? Nicotinic acid, glutamic acid. This is an amino acid source. This is what? Sucrose. It is a carbohydrate source. Okay. So thiamine is the only vitamin source. Another simple question. Simple question. Okay. Which one of the following statement is most correct in DNA replication? They are asking which of the following statement is most correct for DNA replication. They have given here different enzymes along with their function. Kindly match and let me know the answer. Helicase enzyme separate the two strands. DNA gyrase helps in opening of DNA double helix in front of the replication fork. TUS protein helps in termination of replication. Whether this all are correct? Second, option B. DNA polymerase enzyme separate the two strand. This is wrong. Yes, option A is correct answer. Yes, helicase enzyme separate the two strand. This is correct. DNA ligase helps in opening of DNA double helix. This is also wrong statement. Okay. DNA ligase enzyme separate the two strand they have given. This is also wrong. So the right is option A. Yes, option A will be the right answer. They are asking here termination or stopping codons. What are the different stop codons? Okay, we know 3 stop codons are there. Okay, 3 stop codons are there. Okay, UGA, UAA and UAG. Okay, these are the 3 stop codons. Okay, yes, UAA, UAG and UGA. Okay, the most important thing in the remaining option what you have to observe means UGG they have given. Okay, UGG. What this UGG will code? Any guesses? What will this UGG will code? This I have told in replication, transcription, translation class. UGG will code for yes, I am expecting the answer from students. UGG will code for tryptophan. TK. This is very important. UGG because only 2 amino acid have single codons. TK. Yes, only 2 amino acid will have like single codon. One is methionine. TK. Another is tryptophan. TK. Only 2 amino acid have single codon. TK. You have to remember this point also. TK. Next, about tissue culture only. The chemical used for encapsulating somatic embryo to produce synthetic seeds. If somatic embryos are there, what are the chemicals that are used to encapsulate to form this as synthetic seeds? Yes, excellent. Sodium alginate. This is the common used chemical for encapsulating of the seeds. Yes, embryo. Excellent, students. The important features of Seil-Dahlgren sequence. Seil-Dahlgren sequence. This is most repeated. They have asked in 2024 and 2025 also. What will be the correct answer? I know this you will answer. Determines transcription initiation sites. No, this will plays role in translation only. So this will be wrong. Distant mean of determining the translation start site in prokaryotes. Okay, translation. Yes, this will come because the Seil-Dahlgren sequence, okay, that is a purine-rich sequence present in mRNA. This will act as a site for ribosome binding. Okay, so this will come complementary to the part of 3' end of 16S RNA. Yes, this is complementary only. Yes, termination recognition in prokaryote. This also will not come. So yes, option A will be the right answer. B and C. TK, clear? Identify the nucleotide cap. This is another important question. TK, identify the nucleotide cap that is attached at 5' end of mRNA. TK, they are saying what is the nucleotide cap attached at 5' end of mRNA. You have to see here. TK, this is mRNA. The mRNA will have what? Yes, 7-methyl guanosine residue will be there. TK, another important question what they can ask from this is there is an unusual 5' to 5' triphosphate linkage will be there. Okay, you see here, there is an unusual 5' to 5' triphosphate linkage will be there between the 7-methyl guanosine and our target mRNA. Okay, another thing what they will ask means, what is mean by cap 1 and cap 2? This also they can ask. Cap 0 means 7-methyl guanosine attachment. Okay. Cap 1 and cap 2 means the further nucleotides present downstream of this 7-methyl guanosine, the 2' hydroxy will be methylated. Okay. This is known as cap 1. Cap 2 means further next nucleotide. Okay, clear? So, cap 0, cap 1 and cap 2. This is another important concept. Okay. DNA ligase is a molecular glue or gum, yes, which joins together the cut DNA ends of DNA by creating which bond? Which bond it will create? Hydrogen bond, N-glycosidic bond, phosphodiester and phosphodiester bond. Which bond it will create? Yes, phosphodiester, yes, very good. It will create phosphodiester bond between the nucleotides, so it will join the end of the DNA fragments. Okay. The first crop plant genome sequence. What is the first crop plant? Okay. Crop plant genome sequenced. Wheat or rice or barley, tomato. Yes, excellent. Rice. Okay. You have to go through about the genome size also. Okay. What is the genome size for each and every crop? This is also one of the most important point they can ask. Okay. They will give different crops along with their genome. We have to match that one. Okay. This is another important point. Okay. Batch culture, tissue culture. Third question from tissue culture. Batch culture or type of suspension culture where in batch culture what we will do? Whether the media will be replaced every day or the media will be continuously replaced, or it a closed system and medium is loaded only at the beginning, or no depletion of medium throughout the growth period will be there, or cellular waste are continuously removed and replaced. What will be the right answer? In batch culture, what we will do? This is a batch culture, TK. Batch suspension culture. Yes, very good. It is a closed system. Yes, there will be a no replacement of media will be there. It's a closed system and media is loaded only once at the beginning. Very good. Okay. The genes present in Bolga 2 cotton. What are the genes? Bolga 2 cotton. What are the genes will be there? Yes, Cry1 AC and Cry1 2 AB will be there. This EPS will not be there, FA will not be there. Barnes bar star genes, where this will come? Barnes bar star. This also we have discussed in our class. Barnes bar star. Fertility, correct. Yeah, male sterility. We have discussed this in male sterility. Okay, Barnes bar star line. This was isolated from Bacillus amyloliquefaciens. This also we have discussed. Yeah, male sterility, Bacillus amyloliquefaciens. That is the source for this bar star gene. Okay, this is another important point. Okay, T1 generation plant for Cry1 AC gene showed 3:1 segregation in T1. Okay, for selected traits, when the three plant within the gene were selfed, which one of the following statement will be correct? When these three plants are selfed, what it will happen? What it will happen? Whether the two of the three plants will produce all the progeny with the gene, or all the three plants will have that gene, or only one out of the three plants will produce. Yes, very good. Only one out of the three plants will produce all the progeny plants within the gene. Okay, so genetics, from Mendelian genetics, they are sure they are asking 2 to 3 questions. Okay, crossing over like this question. Okay, other thing, other most important question, this we have already discussed in our class. Okay, Bacillus thuringiensis will have endotoxin. Okay, that we know endotoxin will be there. Okay, this endotoxin is gamma, delta, or alpha, or beta. Yes, it is a delta endotoxin. In class itself, we have discussed. This will be active at alkaline pH. Alkaline pH. Okay. What it will do means it will membrane lysis will happen. Okay. Membrane lysis will happen. Because of that, larva will die. Okay. This we have seen. This is a class of endotoxin, delta endotoxin. Okay. Next question. The enzyme that displays histone octamer. We know histone octamer is made up of H2A, H2B, dimer of H2A, H2B, H3 and H4. Okay. This we already know. The enzyme that displaces the histone octamer during transcription. Okay. For example, this is an histone octamer. This is the DNA wrapped around this. Okay. During the transcription process, which enzyme will displace this nucleosome apart? Yes, what it will be there? Whether it's a DNA ligase, no, this will come in DNA replication. Gyrase also will not come. Yes. So RNA polymerase, helicase, what it will do? Double stranded will be converted into single stranded. There helicase will come. So RNA polymerase is the enzyme that will displaces the histone octamer during transcription. Read the question properly. That's why I am drawing this diagram. Okay. The enzyme does displaces the histone octamer during transcription. Okay. They are not asking which enzyme is separating the double strand of the DNA to single strand. Okay. There comes the role of helicase. Okay. To maintain the single stranded, here single strand binding proteins will bind. Okay. Clear? That is other role of SSB. Okay. Clear? Yes. A type of B lymphocyte that produce antibody. Okay, from last 3 years of question paper, only one question they have asked from immunology. Okay, this is not there in our syllabus. Okay, immunology is not there in syllabus. Okay, you don't want to worry. Immunology is not there in syllabus, but in 2023 they have asked one question. Okay, a type of B lymphocyte that produce antibody is what? Erythrocytes. It's a RBC cell. This will not produce adipocytes, fat storing tissues. Plasma cell. Yes, plasma cell is the cell that will produce B-type lymphocytes. TK. Clear? Clear? Immunology is not there in our syllabus, so you don't want to worry. TK. Here, this is the most repeated question. 2024 also they have asked the same question. TK. Here also same question they are asking. TK. Orthologous gene. This we already seen. It will be a homologous gene found in different organism. TK. Homologous gene found in different organism. Splicing, yes, removal of intron. So this will be the right answer. Okay, this will be the right answer. Mass spectroscopy for protein fingerprinting Uniport for protein database. This is we know. Okay. Then listen another question properly. DNA sequencing. If DNA is there, that is sequenced. Okay. DNA sequencing was done. Okay. Sequencing followed by genome annotation. The sequenced DNA was annotated with genome. Are the steps of which genomics? Whether it's comparative genomics, functional genomics, transcriptomics, or structural genomics. Okay. What is comparative genomics? We will compare the structure of the gene or structure or whatever it may be, the elements, whatever it may be, that we will compare between the genomes. Okay. That is comparative genomics. Functional genomics, what? The proteins, protein-protein interaction, that we will study. Transcriptomics, gene expression, particularly mRNA, we will see there. So in structural genomics, they will do the sequencing followed by genome annotation. Clear? The intercalating agent used as a stain for visualizing DNA under UV spectrophotometer is what? This is what? Bromophenol blue or ETBR or silver nitrate or X-gall. What will be the right answer? Yes, this is one of the simplest question. Yes, very good. Very good, very good. Okay. So double stranded DNA will be there. Here bases will be there. Okay. So this ETBR will act as an intercalating agent. It will go and bind here. Very good. Okay. ETBR will go and bind here. Okay. Then what are the remaining option? Bromophenol. Bromophenol is an what? It's an tracking dye. Take a tracking dye. For example, this is our gel. We have loaded the DNA. Okay. We can track the position of the DNA where it is there by using this bromophenol. Okay. Silver nitrate. This is used for silver staining of proteins. Okay. Then what is this X-gall? This will act as a substrate. This X-gall will be a substrate. Okay. This we will be primarily we are using in where? This primarily we are using in cloning. In cloning selection strategy, we will use this one. Okay, because blue color substrate will form, na? Colorless substrate will be converted into blue color. Blue white screening, there this X-gall will be used. Okay. An example of a scoreable marker. In this question paper, they have asked two questions on scoreable marker. Okay. Example of scoreable marker in genetic engineering. What will be the example here? Whether HPT, hygromycin phosphotransferase, ampicillin, neomycin phosphotransfer, or GUS. Yes, very good, excellent. GUS will be an example. Okay. Remaining are what? These all are screenable markers. Okay. Antibiotics, screenable markers. The outer layer of primary plant body which protects the underlying tissue. This is simple question. Yes, outer layer of the primary plant body which protects the underlying tissue is what? Yes, excellent. It is an epidermis. If you remember, in 2024 they have asked about xylem and phloem. What are the vascular tissues? So xylem and phloem. So from the options only they are taking more questions. Okay, we have to focus about the remaining options also. Okay, so epidermis is the outer layer of the primary plant body. Xylem and phloem is the vascular tissues. Okay. The correct order of light reaction. This is also repeated question. The correct order of light reaction. We know. Okay. First PS2 will be there. From here, PO15 will take the electron, accept the electron. Plastoquinone will be there. Cytochrome B6F, plastocyanine, PS1. Here, phylloquinone will be there. This is very important. Okay. Phylloquinone. Phylloquinone will be there. From there, it will pass to where? Iron sulfur clusters will be there. From here, NADH will be produced. Clear? Ferredoxin will be there. From there, NADH will be produced. Yes, this is the sequence. We know PS2 from where? How the electron is flowing from PS2 to this one. Okay. Now, see, first start with PS1. PS2. So, option B. Okay. From there, it will go to plastoquinol. So, B, D, then C, A. B, D, C, A. Option A. Yes, very good. The end product of anaerobic respiration. Simple and direct question. What will be the end product of anaerobic respiration? What will be the end product of anaerobic respiration? Whether ethanol, pyruvate, 3-phosphoglyceric acid or glycerol. What will be the end product? Yes, very good. Ethanol. Ethanol, then lactic acid. This is also end product of anaerobic respiration. Okay. The number of ATP molecules produced during anaerobic respiration is how much? 30, 32 to 36. What will be the answer? Yes, very good. Under anaerobic respiration, only 2 molecules of ATP will be produced. This is 2019 JRF question. Okay. 2019 JRF question. Yes. Yes, yes, very good. Simple question and repeated question also. Different hormones they have given, different responses they have given. I know you people will answer this. Gibberellins, what it will do? Primarily, it will involve in what? Activation of alpha amylase during seed germination. So, one will come option 4. Yes, very good. One will come option 4. Hmm? Correct. Yes, one will come option 4. Indole 3 acetic acid, what it will do? Indole 3 acetic acid. It may be a cell division. It may be cell division factor. It will be there. The most important thing you see here, abscisic acid. This we already seen. It's an inhibition of vivipary. Inhibition of vivipary. This is most important objective. Okay. Abscisic acid will inhibit vivipary. So the right option is what? 4, 3, 2, 1. Clear? 4, 3, 2, 1. Okay. A copper containing, while taking class itself, I told. Copper containing protein that takes part in electron transport in chloroplast. Takes part in electron transport in chloroplast. Cytochrome C. Copper containing electron transport in chloroplast. Read the question properly. Yes, chloroplast means what? Plastocyanin in chloroplast. Cytochrome C oxidase, that is also copper containing only. Copper A, copper B will be there. Clear? Copper A, copper B will be there. But that will be present in mitochondria. Clear? Clear? Just comment yes or no. Clear? So you have to read the question properly there. Okay? Yes, clear. Yes, okay. The chemical used to induce uniform flowering in pineapple. Okay. If we want to induce a uniform flowering in pineapple, which chemical we have to use? Okay. You just observe here. Paclobutrazole, it's a GA inhibitor. Yes. Naphthalene acetic acid, it's a oxine compound. Transietin, this is a cytokinin compound. 2,3,5 trichlorophenoxyacetic, synthetic oxine. Okay. Now you have to answer me. What will be the correct answer? Which can be used to induce uniform flowering, particularly they are asking in pineapple. Okay, particularly they are asking in pineapple. Any guesses? Any guesses? Any guesses? Naphthalene acetic acid can be used. Okay, naphthalene acetic acid can be used to induce flowering in pineapple. Clear? Clear? What this paclobutrazole will do? This is an inhibitor of gibberellic acid biosynthesis. This will also induce flowering. Okay, but particularly they are asking in pineapple. This is specific question. Auxin will induce flowering in pineapple particularly. Okay. Next, extent of canopy cover at a particular development stage of crop is termed as growth parameter. Okay. Leaf area index. Okay. The other definition for this one is this one. Okay. Extent of canopy cover. Yes. Extent of canopy cover at a particular development stage we can say as leaf area index. Okay. This is dimensionless. Okay. dimensionless. Okay, this is also another important point you have to remember. Okay, specific leaf weight, this will say thickness about the leaf. Okay, thickness of leaf can be determined by specific leaf weight. Leaf area duration, this will say stay green trait, stay green. Okay, how much time the leaf is like green in color, how efficiently photosynthesis is happening. So we can say stay green trait can be determined by leaf area duration. Okay. The portable instrument used to record photosynthesis in plant. Simple. Select the option by elimination. What is the portable instrument used to record photosynthesis in plant? Option C, IRCA, yes. Infrared gas analyzer. Infrared gas analyzer is primarily used for determining the photosynthesis activity. Apart from that, here we can take the rate of transpiration, water use efficiency, everything we can calculate in this IRCA. Okay. Remaining C's, if you see, AAS, IPC, OES, this is primarily used for what purpose? For nutrient or mineral elements. Mineral element profiling can be done, correct? So mineral element profiling can be done. Here isotopics will be profiled. Okay, so the right option is IRCA. The typical earliest symptom of iron deficiency, we know iron is what? It's immobile. Okay, if it is immobile, symptom will be present where? In which leaf? Whether younger or older leaves? Yes, very good. Symptom will be present in young leaves. Okay, it's immobile, so symptom will be present in young leaves. Remaining option they have given older leaves, so this will be the right option. Apart from that, calcium is immobile, sulfur is immobile, boron is also immobile. These are the some of the typical immobile elements that are in plant nutrition. Okay. An example of perennial plant that exhibiting monocarpic senescence. Whenever I wrote the JRF exam, they have asked this question. 2019 question, this one. An example of perennial plant exhibiting monocarpic senescence. First, you have to know what is monocarpic senescence. Okay. This will have a growth period. Okay. It will flower only one in its entire life period. After that, it will become die. Okay. So the right answer is bamboo. Okay. This is very important. Okay. Repeated question also. Okay. If you want to practice topic wise mark tests, you can kindly enroll in our mark test bundle. If you want to prepare for ICAR 20, those who want to prepare for ICAR 2027 entrance examination, they can kindly enroll in our next batch. Okay. For first 100 students, offer is also going on, kindly enroll. Okay. Grain yield by biological yield into 100 is what? Grain yield by biological yield, whether it is an harvest index or RGR or CGR or net assimilation rate. What will be the answer? Yes, very good. Harvest index we can also say as EB. Okay. Economical yield by biological yield into 100. Okay. That is economical yield is what? Our grain. So grain yield by biological yield into 100. This is homework for you. Just you go and see what is the formula for RGR, CGR and NAR. Okay. This is homework. Just you go on to see it. Okay. You search by yourself, then you will get more interest for this subject. Okay. You just go on search that one. Okay. An example of non-climacitric fruit. What are the example of non-climacitric fruit? Non-climacitric fruit. Mango, yes, it's a type of climacitric fruit. Banana, climacitric fruit. Avocado, also climacitric fruit. So citrus is a typical example of non-climacitric fruit. Excellent. Manikankam, excellent. Then grapes, lime. These are typical example of non-climacitric fruits. Okay. You have to remember. Okay. Non-climacitric means what? Ethylene doesn't have direct role in fruit ripening. Okay. Even if you apply ethylene, external ethylene, even if you apply, this fruit will not be ripened. Okay. The common chemical agent used as preservative solution to improve the keep quality of cut flower is, this I have told in ethylene class itself. Okay. Silver thiosulfate, STS. Silver thiosulfate, it's an ethylene action inhibitor. Okay. It's an ethylene action inhibitor. It will decrease the activity of ethylene. So, ethylene production will be decreased. Okay. Petal senescence can be reduced. Okay. So, the quality of the flower can be increased. This is very, very important important objective. Okay. Silver thiosulfate. Okay. Silver thiosulfate. And herbicide which belong to synthetic auxin. 2,4-D is the synthetic auxin. They are asking what is the trade name? Yes, any guesses? They are just indirectly they are asking what is the trade name for 2,4-D? 2,4-D. Yes, very good. Dicamba. Take a dicamba. Then I forgot to say in our last class, I told PS2 inhibitors, PS2 and PS1 inhibitor. PS1 inhibitors, paraquat and diquat I told. Okay. This paraquat we can also call as methyl vilogen. This is 2021 question. They have given in the option methyl vilogen. Okay. This PS2 will be inhibited by DCMU I told. Okay. One more inhibitor is there, that is atrazine. Okay. Clear? So, atrazine will inhibit PS2. Okay. Thiodiuzeron, what it is? It is an synthetic cytokinin. This also we have seen. Synthetic cytokinin act as a defoliant. Okay. This is glyphosate for glyphosate. Okay. Glyphosate. Okay. Glyphosate. What it will do? It will inhibit EPSPS enzyme. Okay. Aromatic amino acid biosynthesis will be inhibited. I hope this is clear for everyone. Okay. The amount of water transferred by a plant divided by the amount of carbon dioxide assimilated. See here, amount of water transfer, right, by amount of carbon dioxide assimilated. What it will be the answer? Whether it's transpiration ratio or what? Yes, excellent. It's transpiration ratio. Okay. If water use efficiency means this just will become inverse. Okay. CO2 assimilated by water utilized. Clear? This is most repeated question. Every year they are asking one question from this. Okay. Transpiration rate and water use efficiency ratio. Okay. Quantum efficiency, that we know, number of photons required to release a molecule of oxygen is known as quantum efficiency. Quantum yield means what? Number of oxygen molecule released per photons. Okay. That simple one only. Okay. The process of discharge of liquid from tip of the healthy leaf under humid condition is called guttation. Yes, they have given guttation. Okay. And this process occurs through specialized pores. What is that specialized pore name? Yes, excellent. It's an hydrothode. This hydrothode we can also call as water stomata. Clear? Water stomata. This can also they can give in option. Water stomata. Okay. The experiment help researchers in understanding of photosynthesis in plant. You just let me know. We know Joseph Priestley, what he did, John Ingnos, what he did, Van Nile, T.W. Engelman, what he did, everything. This question they have asked in 2025. Experiment name was different. Okay. What he has predicted? Action spectrum. Action spectrum. Okay. At which wavelength the photosynthesis will be higher, like that he has determined this one. Van Nile, what he did? Yes, excellent, Uday Lakshmi, excellent. Van Nile is the right option. See, Van Nile, what he has predicted means the oxygen released after photosynthesis or during photosynthesis comes from water, not from carbon dioxide. This is the experimental outcome for the CB Van Nile. Okay. This they can ask. We know carbon dioxide will bind with water This will lead to formation of carbohydrates plus water molecules plus oxygen will be released. This oxygen is coming from water, not from carbon dioxide. This is the typical experiment was done by Van Neel. Okay. Ignos, what he did, sunlight is essential for photosynthesis. Sunlight is essential for photosynthesis. That person has given this one. Okay. Clear? So, this is the right option. The moisture content, 2025 question this one. The moisture content in perishable horticulture produce with a short shelf life is, how much it will be there? 3 to 8%, 80 to 90, yes, this we already discussed. So 80 to 90% of water will be there in highly perishable product. See here, so much times they are repeating the same questions. Okay, so you have to be practice much on the previous year question papers also. Okay, okay. The condition required for transpiration, that means the condition required for transpiration, water move through the plant from soil to air. If a water is moving from soil to air, okay, how the water potential should be there? The water potential of soil should be less or greater, or root should be more, or leaf or atmosphere. Just tell me the answer. Yes, we know that water will always move towards more negative water potential, more negative water potential. So it has to reach to atmosphere, means this should have the less atmosphere, less water potential should be there. So if you see, the right option is A. Water potential of soil should be greater than root, leaf should be greater than atmosphere. Yes, very good. Okay. Simple, just you observe, just you use your brain here. It's just simple concept. Harvesting of farm produce, harvesting of farm produce. Okay, see the options: quality loss of harvest produce, quality loss of produce, the technical activity of post harvest system, economic activity of the post harvest system. If you are harvesting some of the produce, it is an what? It's an economical activity. Yes, correct. The marketing means what? Marketing means it's a technical activity of post harvest system. Okay. Loss of seed viability. So first you see, you by first option itself we can easily fill out the answer. Okay. A. Okay. Just a minute, students. Harvesting of farm produce. Okay. They are saying harvesting of farm produce, marketing of farm produce, loss of physical substance, and loss of seed viability. These are the 4 questions they are asking. Okay. They are asking the description of the post harvest system they have given here. Okay. When the seed viability will be lost, when the physical substance will be lost, first we have to solve this one. Okay. When the seed viability will be lost and when the seed physical substance will be lost. Okay. Seed viability, when it will lose, it is whether quality or quantity. Seed viability is just a quality. Okay. So yes, very good, very good. D will come option 2. D will come option 2. So 3, 4, 1, 2 is the right option. Okay. Loss of physical substance. Okay. Loss of physical substance is quantity. Okay. We have solved like this. Okay. Clear? This is simple question only, just you do by elimination method. So option D, A will be the right option. Okay. This is another important question. Okay. This is another important question. They can just see this 75 meter, 1 meter, this question is repeated. Okay. They are saying about water potential so much time the same question. Okay. Every year they are giving different problems. Okay. Different mathematical question. How you see here? In trees, if it is have 75-meter height, the height of the tree is 75 meter. The magnitude of gravitational component of the potential in the leaf will be how much, like that they are asking. Okay. First you have to know what is the component of water potential. Solid potential plus pressure, gravity, matrix. They are particularly asking about gravitational potential if the tree height is 75 meter. Okay. So what we have to calculate first, the potential, how much potential will be there, like that we have to calculate. Formula is there. Okay. So we are doing calculation for water. Density of the water, density of water into gravitational of water, okay, into height of the plant. This is the general formula. Okay, you just remember this one. Okay, density of water is what? 1000 my meter cube. Okay, gravitational potential, how? 9.8 will be there. Height of the water is 75. You just multiply all these things. Okay, it will come around 75 something. It will come. Okay, this will come around 75. Okay, this will come in Pascal. Okay, this will come in Pascal. If we convert the Pascal to megapascal, 1 megapascal equal to 10 power 6 pascals. Okay. So the right option is 0.75. It will come. Okay. Just it's a simple thing. Okay. You have to multiply the gravitational potential with density of water along with the height of the plant. Then we will get the answer in Pascal that we have to convert in megapascal. Okay. An example of single membrane cell organelle associated with oil body in plant cell. An example of single membrane cell organelle associated with oil bodies in plant cell is what? What will be the answer? Whether it's a peroxisome or lysosome or vacuole or glyoxysome. What will be the answer? Yes, I want answer from you guys. What will be the answer? Peroxisome will come or glyoxysome or vacuole or lysosome? What will be the answer? Yes, option A, B, C and D. What will be the right answer for this question? Peroxisome, no. Glyoxysome, you see glyoxysome. Why glyoxysome? We know oil bodies will be there in germinating seedlings. Okay, oil bodies will be there. This oil bodies will have enough of triacyl glycerols. Okay, this will enter beta oxidation. Okay, so much of acetyl CoA will be produced. This acetyl CoA can be converted into succinate by glyoxylate cycle. Glyoxylate cycle in glyoxysome. So, oil bodies will always associated with this glyoxysomes. Okay. Next, enzymes of HMP shunt pathways are located in. HMP means what? Pentose phosphate pathway. Where the enzymes will be located for pentose phosphate pathway? Yes, primarily it will located in cytosol. Okay. But if you see our nitrate metabolism class, I have already told in roots plastid have the enzymes for HMP pathway that will give the NADPH for nitrate reduction. Okay. So in plastids also it will be there, but mitochondria and peroxisome it will never have the HMP pathway. Okay. So A and B will be the right option. Yes, this is clear for everyone. Okay. Next, they are given different scientists and their discovery. Okay. Different scientists and their discovery. Okay. Mitchell, Peter Mitchell. Okay. Chemosmotic theory of ATP synthesis was given by Mitchell. So yes, A3. Okay, fixed diffusion law. The name itself there, fixed diffusion law. Rate of diffusion was given by Fick's law. Okay. Nernst equation they have done for ion distribution. Yes, ion distribution. So this will come as 4. 3 1 3 1 4 2. Where is coming? 3 1 4 2. Option B. Yes, very good option. B is the right option. 3 1 4 2. The nutrient element for the synthesis of chlorophyll, precursor in the biosynthesis of chlorophyll molecule is what? Whether it will be iron or magnesium or molybdenum or manganese. What will be the nutrient element? Iron or magnesium? Yes, magnesium. Yes, very good. Magnesium will be the, that will be present in the central core where 4 chiral ring structures will be there. So, this is very much essential, magnesium. Yes. The amino acid donor to alpha ketoglutarate during photorespiration. They are specifically asking the amino acid donor to alpha ketoglutarate during photorespiration. This is very important. During photorespiration. We know photorespiration will happen in 3 organelles, CPM, chloroplast, peroxisome and mitochondria. Yes. Yes. If you see, chloroplast, peroxisome and mitochondria will be there. In mitochondria, what it will happen means glycine will form. Two molecules of glycine will be converted into serine. Okay. Two molecules of glycine will convert into serine. Here one ammonia will be released. Clear? So the ultimate source is what? It's a glycine. Okay, glycine will be the source of ammonia during photorespiration. If particularly they are asking about photorespiration, means glycine will be the right answer. Yes. In plant cells, the principal ion that is generically pumped across the membrane in plasma membrane and tonoplast is what? This is simple question. This we have seen in our electron transport chain, mitochondrial, chloroplast electron transport chain, everywhere. What will be the principal ion that is electrogenically pumped across membrane in plasma membrane and tonoplast? B. Yes, very good. Okay. Identify the crops that requiring vernalization. We know vernalization means what? Cold treatment for inducing flowering. What will be the example of crops? Example of crops that requires vernalization. Barley, sunflower, carrot and ragi. Yes, carrot ultimately it needed. Yes. Ragi no need. Sunflower also will not come. That vernalization is not required. So the right option is what? A and C. Where it's coming? A and C. Option B will be the right answer. Yes, option B already answer came. Yes, very good. This is another repeated question 2025, 2024, in 2023 also they are asking the same question but different crops. Okay, different crops. Cotton, this we know Maharashtra is the cotton lead producer. Okay, mustard, this we already discussed in all the three question paper. Rajasthan is the largest producer. Yes. Pigeon pea Karnataka, chickpea will be Madhya Pradesh. Yes. Okay, this we already discussed. This is one of the repeated question. That's why I'm going fast. Okay. So 2, 3, 4, 1. 2, 3, 4, 1. So option B will be the right option. Yes, very good. Okay, this is the right option. 2023 part 1. In morning shift, they have asked the same question. Okay. Most commonly used chemicals to break dormancy in seeds requiring light are what are the components? What are the components? Potassium nitrate, kinetin, thiourea. Yes, very good. Same person asking the same. Same person who answered the question. Now also that person is answering. TK. A and C. TK. Potassium nitrate and thiourea. These two are the typical example of the chemicals that are used to break the dormancy in light requiring seeds. TK. Potassium nitrate and thiourea. 2019 also they have asked this question. TK. Clear? Example of non-climacitric fruits. This you have to work out and you have to let me know the answer. Example of non-climacitric fruits. What are the examples? Lemon, cherry, guava, grapes. What are the examples of non? Yes, we have seen grapes is a non-climacitric. Cherry is non-climacitric. Yes, lemon is also non-climacitric. So A, B and D. Where is coming? A, B and D. Yes. A, B and D will be the right answer. Guava is a climacitric fruit. 3K. Yes, so this is the right option. Rickman-Lang effect. This you guys have to answer. I have clearly told this in our lecture itself. Rickman-Lang effect on the process of aging and remobilization of nutrients is associated with which plant hormone? Yes, very good. Cytokinin. Okay, cytokinin is Rickman-Lang effect. Bolting will come for which hormone? Bolting. Bolting for will come for which hormone? Ethylene or gibberellic acid or abscisic acid? Yes, very good. Bolting for gibberellic acid. Very good, students. Very good. Next, identify the crop with zero or nearly zero CO2 compensation point. CO2 compensation point is zero means ultimately it will be a C4 crop. This is the first concept. Now you choose the answer. What will be the right answer? Okay, sugarcane, maize, foxtail. Yes, this is a C4 crop only. Yes. Pineapple, cam is there, so it will not come. Pineapple here also there, it will not come. Pear millet amaranthus is a typical cam plant. Yes, this also will come. So A and C, they are coming, A and C. Option C. Clear. So they are asking about CO2 compensation point is 0 or near 0 will be maximum for C4 plants. Yes, A and C will be the right answer. This you have to match and let me know. Okay. Different institute, I already told from first class onwards. From institute, they will ask, surely they will ask 2 to 3 questions. They are asking match the following institute they have given and its location. Just match and let me know the answers. National Institute for Rural Development, Indian Institute of Sugarcane Research, National Dairy Research Institute, Central Institute for Jute and Allied Fibers. 2341. 2341. Any guesses? 2341. Okay. Yes, any other guesses? Yes, option C. Yes, same. 2341. Yes. Yes, we can see this is the right option. Okay. National Institute for Rural Development is in where? It will be present in Hyderabad. Yes. Indian Institute of Sugarcane Research. It's in Lucknow. Where the Sugarcane Breeding Institute is there. Where the Sugarcane Breeding Institute is there. Sugarcane Breeding Institute. Sugarcane Breeding Institute is located in SBI. Sugarcane Breeding Institute. This is another important thing. Okay, why I will tell. Okay, SBI is located in Coimbatore, Tamil Nadu. Okay. Coimbatore, Tamil Nadu. Why this sugarcane breeding institute is located in that particular location? This location matters. Okay. We know this sugarcane flowers. We will say what? Arrow. Correct? This is another important point. Okay. The sugarcane inflorescence is known as arrow. Okay. Coimbatore. This place have the typical climatic condition, agro-climatic zone for flowering of this sugarcane. Okay, flowering will happen at that particular climatic zone only. That's why SBI is located in Coimbatore. Okay, that's why breeding state is located at Coimbatore. Okay, National Dairy Research Institute. This we already know. It will present in where? Colonel. Okay, next Central Institute for Jute and Allied Fibers. That will be there in Barakpur. Yes, this also we know. Okay, next question, students. An appropriate statistical tool used to compare the difference among three or more than three groups. What we can use? Three or more than three groups. We can use correlation t-test. Yes, very good. ANOVA we can use for comparing the difference among the three or more than three groups. Okay, analysis of variance we can use. Very good. Okay, you have to then another homework I am giving to you. Okay, correlation. What will be the range of correlation? What is the like regression? Correlation and regression range. -1 to +1 forward, 0 to 1 forward. Okay, this you have to go and see. Okay, correlation and regression. This question also they are asking sometimes. Okay, what is the range? -1 to +1 for what? That is correlation. Regression means 0 to 1. Okay, this you just go and see once. Okay, this is another important thing. Okay, desmotubule. Same question we have seen in 2024 question. Okay, desmotubule in plasmodesmata joining the adjacent plant cell is formed from. If two cells are there, plant cell, desmotubules will be there. This will be formed by which one? It's an endomembrane system. This we have already seen. It's an endomembrane system formed by, yes, very good, endoplasmic networks. Okay, endoplasmic network. Identify the crop plants with albuminous seeds. Albuminous seeds. Identify the crop with albuminous seed. Any guesses? Albuminous seeds. You just imagine the crop along with the seeds. You will get. Okay, albuminous means what? You just imagine, this is very simple question only. Albuminous. What are the crops will have albuminous seed? Yes, castor, cashew, yes, pakka this will have. Yes, wherever A option is coming, that is the right answer, yes. This is the only option giving hay. Okay. Castor, cashew, coconut, sunflower, tomato and papaya will have albuminous seeds. Okay. We can easily eliminate the other options. Okay. Then another question they have asked from seed technology. Okay. During 2023, they have asked another question from seed technology. Okay. This is a law. Okay. As per Harrington thumb rules, ISTA rules, viability of seeds depend on storage condition where for a 10% decrease in seed moisture content, the storage rate of seed is doubled. This is first statement. Okay. Second statement, what they are giving? Yes. Second statement, for each 10 degrees Celsius decrease in seed storage temperature, the storage rate of seed is doubled. Any guesses for this? Seed technology, any guesses? What will be the answer? Harrington, ISTA, viability of the seeds depend on storage conditions. Yes, you just to go and see this one, TK. Both A and 2 are correct only. TK, you just to go and see this one, TK. So, seed technology, they have asked one question, TK. This in our last class itself, I told you have to know about the type of stomata. TK, type of stomata, TK. Type of from this, every year they are asking one question. Every year they are asking one question on type of stomata. TK, stomata arrangement, how it is there. Whether upside more will be there or lower surface of the leaf more will be there. TK, isopotamus means what? TK, pomatogeton means what? Like the different terms they are giving. TK, these all are stomatal arrangements only. TK, you see here, you just like apply your brain also. TK, present here, stomata will be present on lower surface of the leaf only. They are saying stomata will be present only on the lower surface of the leaf. Present more on the lower surface and less on the upper surface. Other cases equally it is distributed. Fourth thing is present only on the upper surface. For example, a plant is growing in water condition. Okay, ultimately the stomata should be present only in the upper side of the leaf. Okay, if you see here, this is the typical plant. Okay, Nilomba and Nymphea. That is the typical plant where stomata will be present only in the upper leaf. Okay, so this is clear. So 1, 2, 3, 4. Okay. Fourth will come option 2. Yes. Okay. Present on lower surface only in cold region grown crops. For example, apple and mulberry. That will have a stomata that will present only on the lower surface of the leaves. So first one will come 4. First one will come 4. This is the right answer. Okay. Stomata will be equally distributed in maize and rice. Okay. It is equally distributed in maize and rice. Okay. Present more on the lower surface and upper on the and less on the upper surface is potato and tomato. Okay. The thing what I want to say is just you go through after this lecture, after completion of this lecture, just to search what are the different types of stomata. Okay. What is the particular term we will use for this and what are the example crops. Just you take a screenshot and just you read that one. Okay. This is very important question. Every year they are asking. Okay. Next, occurrence of indole acetal. This we have seen in 2025 question. Okay. Indole acetal doxamine pathway of auxin biosynthesis is characterized to the member of which family? This I already told. Brassicaceae, okay, Musaceae and Oryza sativa. Okay. Rice, this rice. Okay. These three crops, whatever the crops coming under Brassicaceae, Musaceae, okay, and particularly in Oryza sativa, indole acetal doxamine pathway will be present. So the right option is Brassica. Okay. Clear, students? Any doubt is there? Any doubt is there? Any doubts? With this, we are completing the 2023 question paper, both shift 1 and shift 2. Tomorrow we will start with 2022. Any doubt is there in this today's session? I hope all the discussion is fruitful for everyone. We can see in next lecture. Okay, thank you students. Thank you.